Welcome to, unfortunately, a recorded laboratory introduction. This is the laboratory that we're going to have on the 18th of January, which is our first Tuesday of the semester. And right now, it's the 8th of January. I'm recording this, and right now all the plans are that we would be back on campus on the 18th and that we would meet in lab for an hour, each of you for an hour, to go through some set stations of looking at different aspects of sponges and some pretty cool things to see, and to do that in person. If we had to do things remotely, I have backup plans, and don't worry. Remember that last year we were pretty much all remote for a while, as the vaccinations weren't even available until later in the spring semester. So this is, I hope, the only time that we have a recorded lab introduction. Typically that occurs on a Monday, and in this case it's not about COVID, but it's because we have Martin Luther King, a holiday, on Monday the 17th of January, and we do not have any classes. So normally the Monday before the lab we would do an introduction like this. I want to also remind you that posted on the portal is a chart that you'll find that compares all the different animal phyla, and there are characteristics there that you should fill out. I'm not going to go through that in any detail now, but fill it out based on what you're learning here in this introductory lecture. Okay? So the quiz, the 25-point quiz that will be over the phylum Porifera, will be on Monday, January 24th, which we hope will be, as I say here, our first typical Monday. And on the 24th of January, if we're back in session and everybody's on campus and we're going to classes, and I'm healthy and you're healthy, then we'll all be together for that. And on the 24th of January, right at the start of class, which is 11 o'clock in the morning, we'll start out right away with a written quiz. And on that quiz, there will be 10 multiple choice questions based on the information that you needed to learn based on the to know sheet. Of course, the to know sheet for the phylum Porifera. Also the textbook objectives. And I'll be trying to point things out that help to clarify the information you need to know on the to know sheet, and also a little bit of textbook objectives. That's what I'll be clarifying right now as I go through these things. Then, probably on at least the first time we go through this, there will be additional identification questions, and I think for the first time here I'll make a multiple choice, but in the future you may have to write out answers, and writing out answers requires that you get things spelled correctly. But I'm going to tell you that for this lab quiz on Monday, the January 24th, on that lab quiz, the identifications will be multiple choice. So everything will be multiple choice on that first lab quiz on the 24th of January. Okay. Now, right after that lab quiz on the 24th of January, which will take us about 10 to 15 minutes, then I'll do an introduction of the phylum Cnidaria, which is the second phylum. Those are the sea anemones and jellies that you might be excited to learn about, because we hear about coral reefs all the time. Well, an important part of coral reefs includes sponges. So we'll get to that as I finish up these introductory remarks. Now, I said spelling always counts, and when you do have to spell things, I just want to point out a couple things about spelling. So first of all, here's two words that look pretty similar: brachial versus branchial. These are adjectives describing a part of the body. Brachial, the first one, has to do with your arms. Branchial has to do with gills. Arms, gills. One letter difference. Okay. Another term that we will see and learn about is the term efferent and afferent. Efferent, with just a single letter difference, means moving away from. Afferent means moving towards. Now, you don't have to know the definitions of these terms. Just see my point, that a single letter difference makes a huge difference. And here's another huge difference. You should know that when you fill out a job application or send a letter of application or have a resume or a curriculum vitae, sometimes called a CV, prepared that you really should make sure that there are no spelling errors or grammatical errors or any other kinds of writing errors. And I've, now an old guy, have been involved in many job searches, and I can tell you that there's been job searches where we had over 100 applications, and the whole group of us on the committee that were hiring created two piles, those with spelling errors and other problems on just the writing, and those that didn't. And so right away, we saw spelling errors and writing errors as something that was sloppy, something that looked like you didn't pay attention to detail. Imagine that spelling is a bit like the way you would dress for an interview, right? So if you came into an interview and you have a shirt that should be tucked into maybe pants, if it's not tucked in or partially tucked in, if your belt's just improper, missed loops, for example, if you have a tie or a bow of some sort and it's not tied right, if you have something spilled on your clothing, if your hair's all messed up, imagine how that presents you if you're interested in a job. So that's not going to be a very good thing at all. Okay. Now I want to show you a little bit about what the questions over images might look like. I frequently use images on the quizzes that you've seen on the introductions. So I don't always. Sometimes I use different ones. But here would be a sample for questions maybe on some other phyla that we'll come and look at in the future. Now, this is an animal called a nautilus. Really, really cool critters. They have these spiral shells. And so the question could be simply identify the class, the name of a group. What are these nautilus? What class are these nautilus found in? Okay. Here's another kind of a question. I could ask you to identify a structure. And this is another type of a mollusk, and that's actually the foot of something called a chitin. And these are quite common. If you ever get near the ocean, you're very likely to find these, but that's the belly side. That's the part that's stuck to the rock. Over here, we're taking a look at a part of an octopus. And so the question here, identify the structure indicated by the arrow. Finally, here, identify these parts. So question 14 might be, identify that sheet of tissue right here. It's called the mantle. This is a clam. And here, this is the foot. And that's actually the part that you eat when you eat clam chowder or fried clams. All right, a few more introductory things. As far as rules go for the lab, again, masks, of course, have to be worn. No cloth masks. Go to the syllabus. I'm not going to go through all the details right here. I've got it on the syllabus. I did it on the introductory lectures. So let's not spend any more time on that. But you will be turned back at the door if your mask is not proper. If I can't see what it is, if you have a cloth mask over a surgical mask, I'll probably ask you about it and you can explain. That's pretty good, by the way. If you have a surgical mask underneath, closest to your face, covered by a cloth mask, that's actually pretty good, okay? It helps to hold the surgical mask against your face. But just a cloth mask, that's not okay in the zoology lab. Here, you could stop the video. You can see all sorts of other rules. We don't take photographs unless you've asked me, and please stop and look at this. So stop right here and read all these. All right, moving on. Here's our introduction. We're looking first at one of the earliest forms of animals. The earliest animals to form, probably somewhere about 580 million to 600 million years ago. And although we are only looking at, in the labs, 8 animal phyla, I want to make sure that you understand that there are somewhere about 35 animal phyla. Sometimes we disagree on what's a new phylum, and sometimes we find new things. So we're going to look at the 8 that are some of the biggest, but please know there's at least 4 times as many, more than 4 times as many, because 4 times 8 is 32, 4 times as many animal phyla that actually exist. So these are sponges over here, and these are sponges down here. What are the characteristics, the main characteristics of sponges? First of all, they're multicellular, so they have more than one cell. They do not have what are called true tissues. So in you and I, we have different kinds of arrangements of cells and some material between the cells that form things like skeletal muscle tissue or an epithelium that covers the surface of your skin, okay? Or nervous tissue that forms our spinal cord and our brain, right? They don't have that. They're more or less an assemblage of cells that aren't in collections of a lot of one cell type. They're a bit more like a fruit salad, okay? You might sit down and eat a bowl of peaches, or sit down and eat a big bowl of mandarin oranges or pineapple. That would be like a tissue because it's all a lot of the same. But fruit cocktail has just a little bit of everything else kind of jumbled together. So the no true tissues is a bit like fruit cocktail. And after all these years, that's the first time I made up that analogy. So look at that, guys. The last time I teach this thing, I'm still coming up with new stuff. I'm so proud of myself. Body symmetry. We haven't talked about body symmetry. Body symmetry has to do with whether or not we can cut an animal or, do it in theory. And does it create mirror images, right? So right now in you could cut yourself. A person could be cut right through the tip of your nose and in between your nostrils. So between your nostrils, not hitting a nostril, but that little tissue that's between your nostrils, cut right through your belly button, cut right through your anus, and of course openings for the urinary tract, so-called the urethra, and other things that are right along our middle. That makes us bilaterally symmetrical. And we'll talk about different types of symmetry later on in the class. Well, it turns out sponges sometimes just take on the shape of wherever they live, and we'll see that a little bit in lab. I'll show you some real examples of sponges that clearly were wrapping themselves around maybe a rock or a stick or something that had fallen in the water. So their characteristic is they don't usually have a typical symmetrical pattern, okay? And so there, you can see some sponges that look, I don't know, maybe like shrubbery or something that's growing up. Of course, they're all in water. So here's a sponge. It's called a red ball sponge, and they sometimes are actually spherical, but this one's growing against something else, and so it's changed its shape. This organism, this sponge, has got more of an actual symmetry, but it's not typical of what we see in sponges. Where do sponges live? Well, all sponges live in water. All sponges live in water. And we're going to learn that the way they make a living is by actually causing water to move from the outside of their body through the walls of their body and then out the top where there's kind of a chimney. And we'll look for that. They all live in water and they filter water, and that's where they get their nutrients, and that's also what they rely on to reproduce. Most sponges by far live in the ocean. And so those are called marine environments. I don't know if you know that word, but you have to know this. So all that I'm covering right now, you do have to know. Those are marine environments. Well, what about freshwater? Are there freshwater sponges? Yes. Wow, where do you have to go to find a freshwater sponge? Illinois. Here in Illinois, we have freshwater sponges. We have them throughout the United States, and one particular kind of freshwater sponge is called spongilla, and it's a very common sponge to see in the United States. I've collected them in Illinois. I've also seen them in freshwater rivers in Maryland, for example, and other places. In fact, one year I was fishing in a freshwater river by my father-in-law's house, and I was fishing for bass, and I caught a stick, and I brought in the stick just to get it off my hook. And as I picked up the stick, I felt it crunch. Crunch. Now you might think sponges are all spongy. Turns out many sponges are crunchy, like a cracker, not like a potato chip. That's brittle. They're spongy like a cracker. So if you took a saltine cracker and you pressed it between your fingers, it would crunch. And that's what this sponge did. So, seeing that, I brought the stick back to my father-in-law and showed him. And he told me that that was one of the rare times that he was really impressed by something that I knew. In any case, it was about sponges in a river he grew up on, and he didn't know that there were sponges in that river. This is one form that you might see of spongilla, but like I said, when I saw it, it was growing on sort of a flat surface. And also notice how we properly write a genus name. So hopefully in high school, and maybe if you had botany, you learned how to properly write a genus. A genus of an animal, so our genus, so what is the genus of humans? You know? Our genus is Homo, H-O-M-O, okay? And when you write that genus, it's capitalized. And when you write the genus for spongilla, and you don't need the question mark, that's just for the sentence, here's the genus of spongilla. And these terms, scientific names, are foreign phrases. And in formal writing, when you write a foreign phrase, you set the foreign phrase in a different font. You know fonts like New York and Times, Courier. There are many other kinds of fonts that you can see on your computer. Well, when you write it out by hand, you can't put it in a different font because, I mean, I guess you could, but people don't. So instead of putting it in a different font, you underline it. So you underline a genus or you underline a full scientific name. We'll get to that later if it includes the species designation. So right now you need to know it as spongilla. You need to know it's capitalized because it's a genus, and you need to know that it needs to be underlined. Okay? And you always need to know how to properly write a scientific name, no matter what it is. Okay? All right, you ready to learn about smoking sponges? Probably not sponges that look like SpongeBob. But let's talk about actual sponges that are sometimes said to be smoking. So here's a sponge, a commercial sponge, something like what you'll see in lab. And these are spongy. Okay, you squeeze them and they're spongy. Here's a sponge that's more of a tube shape with that chimney at the top. So the water on this sponge, the walls of the sponge have cells with flagella, and those flagella are moving water, and those flagella all around the surface of the sponge cause water here to come into the side of the sponge and up kind of a chimney and out the top. Now, for this picture, I believe that they used a special fluorescent dye, and so somebody had a shot of dye down here at the bottom, and the dye was quickly pulled up into the sponge and then out the top. And so they took a photo so that you could see that water flow. Over here, I believe that this is a normal sponge, and they're called barrel sponges, and this is dinner plate size. And guys, this could be a whole dinner plate. It's huge right here. This is much bigger than a basketball. And I've seen these in the Florida Keys. In fact, it was like a stool, like, you'd sit down on or something. And boy, you have to be careful because they're really, really old. This thing could be over 100 years old. So, of course, I didn't want to disturb it. But here is a sponge that's releasing probably some kinds of gametes. So we call those eggs or sperm over here. So I think that's a natural color right there. Now, something else I just want to toss in. If you ever lift a sponge out of the water, you should consider that you've killed it, because air gets trapped in the walls of the sponge, and then it won't be able to filter like that. So when I was at really low tide in what would have been probably about 5 or 6 foot of water, I was down to my waist and standing near these kind of big sponges. And of course, they were not sticking out of the water because then they would die. But I was bumping into them and realizing, wow, this is a big thing that's pretty deep water, and I must have waded out at low tide to a pretty deep spot. So here's some of the different shapes that we see of sponges. And at this point, aren't you kind of thinking to yourself, these aren't animals? I got some cocoa here I'm sipping. You might think to yourself, these aren't animals. I mean, come on. They're plants. Yeah, Zalisco, I know. I'll write the stuff down on the test. But in your heart of hearts, you really don't believe these are animals. Well, one of the things for you to make sure you understand is they don't use photosynthesis. And so they're not plants. Now, interestingly, some sponges have algae that lives within the walls of the sponge, and so they might look like they're photosynthetic, but the sponge cells are not photosynthetic. Only the algae that might get pulled into the walls of the sponge. So here's some more real living sponges. How do they feed? Here's what I was telling you about before. These cells, called choanocytes, have this flagellum. You can see that these choanocytes are located right here on the inside wall of the sponge. Okay? And so this is one pulled out right here, but there they are with their flagella there. And as they wave their flagella, like a fish tail almost beating in the water, it causes currents of water to move through little tiny channels through the wall of the sponge into that central cavity, and then like a chimney going right out the top right here. So they are filter feeders. And this is the simplest form of a sponge, in which it's just a simple tube with a little chimney at the top. You'll need to learn that the name of that little chimney is called an osculum. Osculum, O-S-C, osculum. It's on your to know sheet. Some sponges are really complicated and they have lots of chimneys, but it turns out houses are the same. So if you look at probably old houses that used fireplaces and chimneys to allow them to have fires and heat the home, there might have been a fireplace in every bedroom. I actually stayed in a mansion in the South one time, in which every bedroom, it was just like Gone with the Wind, kind of a southern plantation house. When I was down there, each bedroom had its own fireplace. And so, like a complicated sponge, that house had lots of chimneys. That's my point. This is figure 33.4, so I'm flashing it up here in the right corner. Figure 33.4 in your textbook. So that's the point about using textbook figures for our lab. Good. Now, classifying sponges is a little more complicated, and I've tried to help you out with the to know sheet. Okay. Sponge skeletons are how we classify sponges. Well, what's the sponge skeleton like? Well, it's not like most of the skeletons you've ever learned about. We have a skeleton that's all together. We talked about the vertebral column, that series of bones that forms our spine. We have an endoskeleton. That means it's on our inside. Insects that we talked about are exoskeletons. Those skeletons are on the outside, like armor that might have been worn on the outside, a shell. Here, the skeleton of sponges are actually little bitty hard things that are all scattered within and amongst the different cells. Okay? And those little particles are called spicules. Spicules. So sponges have spicules for their skeleton. And what are those spicules made of? Well, it varies. And what are the shape of the spicules? Well, that varies too. And so zoologists use the composition of spicules and their shapes to classify sponges. Okay. Sponge classification based on the composition and shape. Spicules might be made of calcium, silica, or protein. Calcium you can think of as like bones, silica is like glass, and protein is pretty much what a steak is or a hamburger is. Okay. So it's flexible. So if you think about this, sponges that have calcium-based spicules or silica-based spicules are probably crunchy sponges. And sponges that have protein-based spicules are probably the flexible sponges that I'm going to guess that you expected. So here's one of the classes of sponges called class Calcarea. And from the name, you could guess that it's calcium-based spicules. And this is a very simple sponge. It happens to be stained green here. It really does look like a plant, but it's a branching system here. And stained green, there's some details of it. You can see the little chimney, the osculum right here at the tip. Here's now some of the arrangements of the cells within this sponge. And here's what its spicules look like. So can you think of a brand of car where it's kind of a, this is the symbol of it? It might be on the hood of a car. Mercedes. Mercedes uses that symbol. So spicules between the cells, and they're made of calcium carbonate in this class. And above that genus is Leucoselenia. And we're going to see the whole sponge, and you're going to look at spicules of it in lab. In fact, one of the neat things about this, and I really hope you get to see it, you can come to lab and see it, and that we're able to come to lab and see it. I really have a microscope slide that just looks like this. But when you zoom in on and use a higher power objective, and remember you have to go through and see those videos, the microscope videos, before the 18th of January, before this lab. You need to know how to properly use my scopes, and I'm fussy about some stuff, and I'm going to yell at you. If you aren't doing it right, I'm going to try to be nice. But I'm going to say, whoa, 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 stop, whoa, no. And we'll talk about what you're doing wrong. And yes, it'll be in front of others, because I'll have to stop you right away while you're at a table. So let's try to avoid embarrassment and learn how to do this. By the way, if you hear a little bit of a whining in the background, there we go. Maybe you just heard that. That's Lucy, my other dog, breathing just beneath me. So don't think that you have a technical issue. It's just Lucy breathing. And you're bound by my feet. We will zoom in on this Leucoselenia specimen. And as we do and get to a higher level, now this is a drawing, you'll actually be able to see all the spicules. We don't have to do any special preparation. It's like an X-ray, but it's not, because you can just see it with regular light. So I think it's really one of the cool things that I like to see in zoology is you can literally see all the spicules in place in a specimen if you use the microscope properly. Okay. Now another sponge we're going to see in the lab is something called Scypha, and it looks fuzzy, and it's maybe an inch or two long, and it's wet, and its walls look wrinkly. So what would be the adaptive advantage? Now I want to start this explanation that you're going to hear me say all semester long. Some people might say, well, why do they have that? Well, in evolution, evolution is not deliberate. Organisms do not get things because they need it or want it. What happens in evolution is that there's different variations in species, and some variations convey an advantage. And if it conveys an advantage, we think those individuals or species are more likely to survive and then also reproduce. And that's natural selection. That's evolution. So when we look at something like this, an adaptation, we don't ask properly the question, well, what do they do that for? Why did they do that? Because they didn't choose to do that. What I say is, what do you think the advantage was of this? What was the adaptive advantage of this? And I don't know if you can think of what would be the adaptive advantage in a filter feeding animal to have a highly folded surface. And it's a real important adaptation. We have this kind of adaptation in us in many places. And the answer is it increases the surface area. There's more surface area. So if you look at your hand right now, and if you make a fist, your fingers are not as much exposed to the air. Now extend your hand out with all your fingers out, and now you have more exposed surface. And so on a cold day, this would lose a lot of heat. So on a cold day, we usually make a fist if we don't have gloves or mittens on. And we're going to talk about that all semester long. So it's an adaptation to increase the surface area, or that increases the surface area. Now another group of sponges are they're spicules, and this is the sponge class called hex. Let's take it apart. Hexactinellida. Hexactinellida. That's the class of sponges. Hexactinellida. Okay. And they're called the glass sponges, and they're some of the neatest things that you're going to see. And I have real examples for you. Their spicules are made of silica, which is essentially glass. And this is what these guys look like. And this is maybe up to 8 to 12 inches tall. And this is a group of them that somebody mounted in a nice little display here. And oh my gosh, are these brittle. This is just, you almost look at them and they crunch. So it will be very rare if you handle these in the zoolab. There may be circumstances, but with COVID, we probably won't. And it forms like a cage here. It's sealed at the top. It's sealed all the way around. And the genus of those, which we'll look at, is called Euplectella. Euplectella. Okay. And so these are glass sponges. And I'm turning away really, really quickly because I'm just curious about something that I'm looking up while I'm recording this. I want to see if you can buy this on Amazon. That's really interesting. You cannot. I thought you could pretty much get anything on Amazon, but you can't. Sponge Euplectella. How about that? Huh? Nope. I thought somebody would be selling these. So, in any case, there are places where you can buy them, and lucky you, I've collected a few, bought a few over the years, so that you can see several of them in lab. Well, here's what their spicules look like. Six-pointed like this, made of silica again, and this kind of looks like it's a jack, maybe. Jacks like people play with. All right. Now, the magic thing about Euplectella is that there's something living inside, and the answer is it's another animal, and it's a big animal, and it's a type of animal that you're well aware of. Okay. So the question is, what's living inside these sponges? Why is it there? And you have to find out as you read through and prepare on your to-know sheets. Okay? So we'll come to lab and you'll find out. You also get to see what's inside. They're dead, but you can see their bodies. Finally, the last group to look at is the group that's commercial sponges. This is the most abundant by far. Class Demospongia. Spicules could be made of silica, so I mentioned to you the freshwater sponge. See if you can remember the genus of the freshwater sponge. The first one I was telling you about that I found in Illinois and in Maryland. And those are called Spongilla. So Spongilla belongs to this group, and it's crunchy, so its spicules are made of silica. But most of the sponges, and the big ones you'll see and the ones you'll be able to squeeze in lab, are flexible, and their spicules are made of protein. So Spongilla is the freshwater one, and the commercial ones that we'll look at, their skeletons made of spongin. Again, you can pause this video anytime to write better notes, so that's really lucky to have this recorded. Now, on your to-know sheet, I created this chart. And this chart, I should have given more room for the last letter here. So that E goes on the end of this, that A goes on the end of this. Here are the three classes of sponges that we just talked about. Here's the type of spicule. So we said calcarea has calcium. Hexactinellida, that was the goofy one that's standing upright with silica. And then we just talked just now about the commercial sponges, the squishy ones with protein spicules, and the freshwater one with silica. Okay. And over here, as you follow the chart, within the class Demospongia, those that have silica spicules is Spongilla right here. And those that had protein right over here, commercial. And we don't have to learn a genus for the commercial, lucky you. Within the Hexactinellida, the example we looked at was Euplectella. And over here within the class Calcarea, we looked at two examples, Leucoselenia and Scypha right here. Okay. Now there's one more aspect of this chart that you can see right here going up and down here. And I can tell that when I'm dragging my mouse, I think you can hear this. Listen. Sorry about that. Nothing I can do about it right now. In any case, there's another aspect of sponges we haven't talked about, and that's the body plan. And in short, it goes from simple, kind of like the hot dog thing we saw with Leucoselenia, a little more infolded that we saw with Scypha, and then leucon is the very, very infolded body plan that down here we saw in commercial sponges. Well, as you can see from this chart, you cannot rely on this body plan for the classification of sponges. Instead, right here, remember, we're using spicules, the type of spicule, for the classification. Okay, right in here. I disconnected my mouse. I just figured out how I could stop the sound. And you can see that the body plans, like the leucon body plan, is found in all three groups right here. So clearly, you cannot use body plan to classify sponges. Okay? But otherwise, this is a heck of a really nice chart to help organize the information, and you're welcome. So here's the body plans. Over here on the left is the ascon, cycon, and leucon. So this is the simplest. We saw this in Leucoselenia. This is the more complicated fuzzy surface. So we talked about the surface area. This is cycon body plan, and we saw this in the genus Scypha. And over here is the highly infolded body plan, often with many different oscula at the surface. And this is what we see in commercial sponges, and this is the leucon body plan. Okay. So most of the classes of sponges have more than one body type, and so we cannot use body type to classify. So here's that chart again that we just looked at. So I don't need to spend a lot of time here. We're done, guys. I want to show you just some funky things. These are a type of a sponge that was described in the last 10 years or so. It's called a lyra, which is like a harp sponge because it kind of looks like a harp. That's what I guess a lyra. Boy, I could have that wrong. But anyway, a lyre, I guess, a lyre sponge right here, just to show you a new one. And yes, don't they look like plants? I mean, it even looks like plants with roots down here, but those aren't roots. Okay, so here's a bit of information from the publication. It looks like that publication was in 2012, so it was exactly 10 years ago that they published this. Finally, just a reminder about where we're headed. Okay, so by the 18th of January, you want to watch both of the YouTube videos on the microscopes. I will correct you. If you're not using the microscopes right, I will not come over and say it quietly because we have COVID. So I'll probably sit far away from you and watch you guys like a hawk. Actually, I'll be watching to see if I can help you and clarify anything, but I will also be watching to see if you're making mistakes here. Finally, what do you need to bring to that lab then? So it'd be nice if you had your word root book. If you found the file online for the word root book, then you could bring that electronically. If you have a laptop, that's a good idea. Bring that. You can look up pictures. And what some people do, they create their own slideshow while they're even in lab or as they're studying by finding images on the internet, maybe some of them similar to what I just showed you. And they'll either use Google Slides or Microsoft's PowerPoint, or if you have something else, and they'll create their own slideshow of all the different things that they need to know, and then they can quiz themselves over that. And so that's been a very common way that people who've gotten good grades in zoology have gotten highly organized. So these are all the things you're going to need to have brought, okay? Also, I'm asking that you just bring something that you'd like to look at under the dissection microscope. So I'll just tell you, a $1 bill, a $5 bill are really, really neat to look at because you'll notice things on money that you've never seen before. If you have jewelry and you'd like to look at details of the jewelry, you'll notice how dirty it is, probably. If you look at your fingernails, it's usually embarrassing because we all have stuff in our fingernails that's embarrassing. But please bring something and be curious about it, okay? One of the most important things that you can do in science is be curious. I hope that for you. All right, we're done. End of slideshow.